Welcome to my poster. So, the human cerebellum is a fairly large area at the bottom of the brain. It tends to be quite underexplored, and the reason for this is that it has a very extensive cortical seat that's very highly folded. So you really need to push the resolution a lot to 400 microns or even beyond to be able to tease all the cortical detail apart. But if you manage to do so, you'll see that there is a lot of detail that's similar to the neocortex. So we have a very consistent layer structure. And similar to the cortical columns, there are also clusters of neurons that tend to run from the anterior to the posterior, that they're called the cortical stripes of the cerebellum. And you can see them here, for example, in a mouse. And these stripes tend to be a consistent functional organization across the vertebrates. So both in mammals and birds, we see them very, very consistently. And it is hypothesized that they also exist and are very important in humans as well. However, we have never quite seen them. And this is because 2D histology doesn't quite tend to be able to see them. Because of the tight folding of the cerebellum, you really need a 3D measure. And with MRI, we straight up do not quite have the resolution and the tool so far. So ultra-high field may be able to help. However, with the cerebellum, we bind to all sorts of issues. First of all, with typical ultra-high field scanners, typical coils, the Bion profiles tend to be very bad. Also, B0 tends to be problematic. Also, if you push the resolution, motion becomes an issue. And finally, we just don't have, honestly, the analysis and processing tools that we need. That's the hole we wanted to plug here in this abstract. And we want to explore whether we can use these tools to image the cortical stripes. So first of all, we set up our motion correction. It's an easy-to-use algorithm based on FAT navigators. So we segment our M2 rates every 5 seconds, and we insert FAT navigators that we use to reconstruct, and then we use to track motion and update our field of view online. So I have presented this algorithm before, also other colleagues as well. It tends to work quite well. The important thing here is that it's actually something that's very easy to use for neuroscientists or for clinical researchers. Having done that, we ported this setup to our parallel transmit system. We still wanted something that's easy to use, so what we thought of doing was to basically look in the cerebellum and try to face him there. And it turns out that if you compare the seams, the face seams, between individuals in these small, relatively small ROIs like the human cerebellum, quite often the solution that you see is very consistent. So the face offsets within the same transit across different individuals are the same. So for example, here you see in blue, one transmit with each dot representing different participants, but you see the solution that you end up getting is the same. So that implies that you can calculate a, so to speak, global RF sim that optimize the cerebellum very easily. Emma Brouwer, my colleague, has shown this before, as has another abstract in this conference that I invite you to check. So by combining this, easy to use PTX, easy to use motion, we can get pictures like this, MP2 ranges at 400 microns that have nice white and gray matter contrast. And you can use this to segment the cerebellum. And it takes some doing, I have to say. It's not easy to do the segmentations, but we have managed by now to almost fully automate this. And we can make cerebellar cortical surfaces that capture a lot of this fine anatomical detail. And I invite you to compare this to a standard analysis. You'll see that we have a lot more detail that's just missing, typically. So having now this very detailed cortical surface, as I said, we want to see if there may be some stripes, so if there are structures like this. So what we did is just an fMRI protocol, a simple motor experiment with finger flexing, indexes, also other fingers. And it turns out, when we project this fMRI activation on the cortical surface, we do see a pattern that looks a bit stripe-like. You see this here. And that's kind of consistent. So we tried different fingers, different protocols, different runs, different days, different participants. In all of them, we could kind of see a pattern like that. Now, the problem is that we're not quite sure whether what we see in bold fMRI is actually neuronal or it relates to the vasculature. And this is even worse than cerebellum because we are actually not quite sure what the vasculature in the cerebellum looks like. So what we did is look into developing arteriograms and venograms. And it took some doing, but we eventually managed to do this. So we had TOF acquisitions, RFC, that after some optimization of the orientation could show us some arteries to the cerebellum that looked quite convincing and similar to what we expect from ex vivo studies. And the same for venograms. Here I have a GIF where I ran through it. The unfortunate thing is, if you notice here on the axial side, you see some black patterns, which are the veins, that are oriented similarly to what we expect our fMRI activation to look like if you have stripes. So that means that it's kind of hard to actually tease them apart from true neuronal activation, the fMRI activation versus venous-related activation. And indeed, if we overlap this venogram to bold, which you see here in red, you see that we have a very tight overlap. So that's an issue, which meant that we had to also look for alternative functional acquisitions. And there are alternative contours that are less vasculature weighted and still can be very specific to neuronal activation, like vaso. And that's a contrast that people haven't used in the cerebellum. It was quite tricky to do so, but we did a variety of tricks. We used our RFSIMs, we optimized the orientation, we optimized the echo time, a variety of things. Then in case, you can contact me for more detail. But the important thing is what you see here in green, which is vaso, overlapping red and yellow, which is bold, shows you that vaso actually works, can produce clusters that are symmetric, bilateral, more symmetric than bold, more specific. And if you project them to your venogram, you see that they do not tend to overlap like bold with the veins so much. So this is a measure that's less venous weighted. And when we project it to our cortical surface, it still has this stripe-like organization. But of course, it's not perfect. It's not a perfect measure, vaso either, especially in the cerebellum, because the bold optimization, the bold normalization, it's not quite perfect. And indeed, when we try to look, for example, to things like histology to validate our findings, what you see is that in mice, it's actually very easy to see these stripes, but in humans, when we tried, not quite as easy, and that's because of the tight folding, but potentially also because maybe the stripes in humans are a bit more complicated than we expected to be. So in summary, we developed a lot of imaging tools that work very well. So sequences, motion correction, PTX side of things, software. We now can do a lot of things in cerebellum that we're not able to do before. But we're still not sure that we have actually quite grasped the cerebellar stripes. We are in the same resolution ballpark than we expect, but I'm still not convinced, to be honest. Still, it's an important target, so I'm glad we tried. Thank you for your attention. I would like to thank my colleagues who did a lot of this beautiful work. And please check the colleagues from our group on ultra high field